Plasmodium falciparum establishes chronic infections through high viral gene expression switching rates.
Bonjour à tous. I had the pleasure of doing my postdoc here 10 years ago, so it's great to be back home. But nowadays, I start all of my talk about climate change, just 1 minute, because I like to share my eco-anxiety with everyone. And today I thought I would present a few key numbers that are important to keep in mind if we want to try to reduce our carbon footprint. Some actions might be more useful than others. All of us should aim for a carbon footprint of less than 2 tons of CO2 per person per year. The problem is that in Europe, the average European already has a carbon footprint of about 10 tons. Average American is twice as much. Average Nigerian is 50 times less. And why is it so big? It's, well, as you know, is partly because of flying. Just a round trip from the US to here is you've already exceeded your quota of CO2. So those of you who have joined online, thank you very much. We appreciate it. Another reason is the amount of meat that we consume. If you eat beef every day, that's already 3 tons of CO2 per year. But that can be reduced by up to 90% by reducing the amount of meat consumption. So that's the sort of numbers to keep in mind when you have to make a decision. Now that I have killed the mood, let's get back to business. As malariologists, always start our talks with the number of clinical cases per year. And of course, the clinical research is always done on clinical samples. But if falciparum was presenting this talk, I think it would say, hold on a second. You're presenting the exception here, not the rule. The rule is that the large majority of all falciparum infection remain asymptomatic. That's the elephant in the room. And surprisingly, very little research has been done on this asymptomatic infection. And they are going to be a real problem for malaria elimination. The last parasite standing will be the strongest. So in our group, we like to see this challenge as an opportunity to study host pathogen interaction in real time of the same infection in the same host over a long period of time by collecting regular blood samples from the same host. And this is why we collected, we organized a study in the Gambia, West Africa, which spanned over 2 years. We aim to characterize this host pathogen interaction from the population level all the way down to the molecular level. For the population level, please go and see Marc-Antoine's poster, especially if you happen to be a reviewer of the paper. And I will focus on chronic asymptomatic infection during the dry season, so that we're recruited at the beginning of the dry season. And I will focus on trying to understand how the parasite is able to evade the immune system for so long. So the textbook is telling us that the parasite expresses surface antigen, and the parasite is able to switch from one antigen to the next to evade the immune system and to force the immune system to acquire new antibodies against each new antigen. And these new antigens are encoded by a family of about 60 VAR genes. They encode for the protein called PFAMP1. And the feature about VAR gene is that they're extremely polymorphic. And each time you sequence a new isolate, typically you find a brand new repertoire of 60 VAR genes. So the total number of VAR genes in the world is virtually infinite. Yet in each individual cell, you find mutually exclusive expression, only one VAR gene expressed at a time. And so we know a lot about those VAR gene switching in vitro, but surprisingly, no one has ever tested whether this model is actually what's happening in chronic long-term infection. So we try to do that with 12 chronic infections that were collected during this dry season in the Gambia. Let me tell you a bit more about the methods first, because working on VAR genes is difficult from a failed isolates because of what I was saying about this VAR gene polymorphism. You can't simply map your RNA or DNA reads on your 3D7 genome, because 3D7 would have a different repertoire of VAR genes. What we had to do was to first culture the parasite in vitro, just to bulk up and get more DNA, sequence that DNA with PacBio technology here at Sanger, thanks to Mara and her team, and from that we can extract the full-length VAR genes from each of these isolates. In parallel with the ex vivo sample, we can extract the RNA and use the DB alpha universal primers. Let me explain. Each VAR gene starts with a DB-alpha domain, and within this domain, you find two conserved motifs, and you can use primers to amplify this polymorphic sequence. And this polymorphic sequence is basically a barcode that is unique to each var gene. And you can then map these barcodes on your full-length var gene, and then voila, finally, you've got your full-length var gene expression at every time point. Here are what the first result look like. I'm just showing one example from one individual, 9-year-old male, 5 monthly time points. Each bar is a var gene being expressed. And as the model told us, we are seeing indeed a lot of switching, limited amount of var genes expressed. So that seems to fit with the model so far. To analyze our data globally, we use an entropy metric. Low entropy means low amount of var genes expressed. High entropy means more heterogeneity in the var gene expression. There is no correlation with parastemia. That's very reassuring because it would indicate that we are able to detect all var genes expressed, even in very low parastemia samples. And this var gene expression entropy is on average lower in the dry season in these chronic infections compared to more recent infection, mainly symptomatic infection from the wet season. So I think this indicates that there is some selection against some var genes within chronic infections. And this relatively low var gene expression entropy is somewhat stable over time, over the 6 months of the dry season, although with variation. So each line is a different host. And perhaps surprisingly, in polyclonal isolates, so isolates with multiple strains, the var gene entropy was the same as in monoclonal isolates. And naively, I thought that, because you have polyclonal infection, that means you don't have 60 var genes, but 120, 180. So I thought we would see more var gene expressed. That's not what we see. So perhaps the immune system tolerates only a very limited of PFMP1 antigens at a time, irrespective of the number of circulating strains. Now, I'll go back to my first example, because there's something surprising we noticed, and it's that some var genes appear to be recurrent. And by this I mean a var gene that is switched on, like this one here, switched off the next month, and then switched back on again. We call it a recurrent var gene. We find 1, 2, or 3 recurrent var gene in most volunteers, in 75% of all volunteers. And clearly, that does not fit with our oversimplified model. So what that means is that we need to go back to the drawing board and find a better model, and trying to explain where these recurrent infections come from. So to recap on the main messages, first we show that in chronic infection, you find a relatively low number of var genes being transcribed, plus some var genes at background level. So that could be parasite switching from one var gene to the next. We have shown that indeed there is switching in vivo as expected, but we find these recurrent var genes, which really these results bring more questions than answers. We would need to know whether these recurrent var genes elicit an antibody response. And if anybody wants to help, I would be very grateful, because there's still a lot of work to be done here. And we need to measure how short-lived is this immunity. Obviously, if we want to measure the in vivo switching rates, it's clear now that we need to sample much more regularly than once a month. And on this, I would like to thank everyone involved in the study, mainly Prince and Mathieu in my group, who did all the work, basically, but also with the help of Thomas Lafson in Copenhagen. I already cited Mara and her team here at the Sanger Institute. And last but not least, all the participants of the study. So if you